Our second video for Chapter 1 covers the scientific method. So this material is actually found in your textbook and in Lab 2 of our lab manual, so you're welcome to refer to those for a little bit more supplemental information. So the scientific method is the way that we study scientific phenomenon. So science is an attempt to understand our world. We are biologists, so we are attempting to understand the living world, but there are lots of different types of science. I'm sure you are all familiar with different types of science. But we are going to be using the scientific method to study biology. So all of our labs are going to be designed around learning something for your intents and purposes related to the tests and related to the topic of biology, but they're all going to be something that has to do with a living system. So in our attempt to understand the world, we often start with asking a question or observing something that interests us. Why does this happen? How does this come about? What is going on? This is really cool. What's causing this to happen? And we call this discovery science, where we are basically looking for an explanation. We're looking for why something happens. What's the cause? What is going on? And we're going to do this using a hypothesis. So a hypothesis is a possible explanation. So we're going to start by asking a question, and then we're going to do a little bit of outside research because a hypothesis is not a wild guess. A hypothesis is actually based on what we do know about the subject that we're looking at. So we're going to talk about how we go through this process of inquiry, observation, experimentation, forming a hypothesis, testing that hypothesis. And then we're also going to talk a little bit about theories, because a theory in the scientific community is a little bit different than you might use it on a normal daily basis. So we'll talk about the differences between a hypothesis and a theory as well. All right, so how do we actually do science? How did we learn all of these things about our natural world? Well, some of it was actual, just good old observation. So just sitting back and looking at what's going on around us. There's absolutely nothing wrong with that. Observing, not asking why, just observing is really important in understanding our world. But if we're going to explain things, if we're going to attempt to find the root cause of something, we want to do the scientific method. So the scientific method starts with a question or an observation, something that you find interesting, something that you're wondering about. And then the next step is an experiment. So the scientific method is trying to use an experiment, a set of parameters to answer your question. And we are worried about causal questions. We want to know what the cause is for the phenomenon that we are observing. And we do this in a very carefully controlled experiment. So I've got this long definition of a controlled experiment here. A controlled experiment is an experiment in which all factors in our experiment group, the one that we are looking at and observing, and a comparison control, so one that's not receiving any treatment, are kept the same except for the one factor that we want to change or alter. So what this means is that you have two groups. You have your experimental group and you have your control group. Now, a control group can kind of be thought of as the normal group or the non-treatment group. Now, you get to decide what is normal and non-treatment, but they are the group that is not receiving any change. The experimental group is the one that is changed by one factor, and that is the factor that you think is the cause for the phenomenon. So we're going to talk about a lot of different definitions here, and a lot of them use the term variable. So as we're going through with this, I would love for you to pause and write out some flashcards that actually differentiate between the different definitions of variables. One of those is the controlled variable. So the controlled variable is this one factor, your experimental factor, that is going to change across your two groups. So when we are designing an experiment, we are going to have our groups. We'll have our control group, and we are going to have our experimental group. Now, you might have multiple experimental groups. We're going to stick with just two right now to keep it simple. But remember, our control group is the group that's receiving no treatment. And our experimental group is the group that is receiving some sort of special treatment. So our experimental variable is the variable the variable that changes. And it is the one variable that we think will affect our outcome. So it is essentially the variable that provides the explanation. If you change this one thing, what happens? This one thing, you're interested in, how does this one variable affect the situation? In contrast, our controlled variable is the variable that doesn't change. So it is the condition that is applied to our control group. So our experimental variable is applied to our experimental group, and our controlled variable, which is usually no treatment or a very set of conditions that we are using as a baseline, is applied to our control group. As far as measuring our data, we actually often refer to this experimental variable as the independent variable. So if we're going to call it the independent variable, it is still the variable that changes. And it is the factor that is controlled by the researcher. So experimental variable and independent variable are essentially two words that mean the same thing. It is the factor that we are looking at as the agent of change. The dependent variable is essentially the data that we measure, and it is caused or changed by the independent variable. So another way to think of this is that our independent variable affects or changes our dependent variable. So our independent variable is going to be the experimental setup. And our dependent variable is going to be the data we collect. Now you're going to collect data on both your experimental group, the one that's receiving your special treatment, and the control group, the one that's not receiving your treatment, so that you can compare the differences between those two groups. As a quick little side note, when we are graphing, we are always going to put our independent variable on the x-axis and our dependent variable on the y-axis. And this is going to allow us to, across all graphs that we make, see the impact of the independent variable on the dependent variable. So we actually don't design our experiment first. I sort of went out of order and explained some words that I'm going to be using in our experimental variable, our control variable, etc. Really what you're going to do is you're going to ask your question. Then you're going to do a little bit of research on the topic and just kind of see what is already known. Are there other systems that this has been studied in? Are there things that you might learn that might affect the outcome of this question? So based on your previous knowledge, your observations, the question you want to ask, you're going to form a hypothesis. So a hypothesis is not an educated guess. It is an educated explanation. So you actually have to know a little bit about the system in order to make a hypothesis. But it is an explanation based on observation and previous known assumptions, other experiments, etc., that lead to a testable prediction or an experiment. So when you are working in this class, I am going to expect a hypothesis that is based on the information that you should know. So information in our pre-labs, information in our textbooks, information on the introduction to the lab, where you are given background information on a particular topic. So sometimes students will say, well, a hypothesis is just a guess, and I guessed this, and you marked it wrong. It's not wrong in the sense that, yes, it could be a possible explanation, but what I'm looking for is for you to not use a hypothesis as just a guess. I want you to look at observations and other information that we're giving you to make a reasonable explanation. Because it's not reasonable if you know that it's going to be wrong based on what you know about the situation. Now, that doesn't mean that your hypothesis is going to be supported. It could be not supported in your particular experiment, and that's okay. I'm not worried about whether or not you support your hypothesis as much as I'm worried about you making a hypothesis that makes sense with the information that you are given. So it's not a guess. It is a plausible explanation. And it also looks for the cause of the problem or the cause of the phenomenon that you're looking at. A few things about a hypothesis. A hypothesis must be testable. So if there's no way that you could design an experiment around that hypothesis, then you can't use it. And then, of course, some questions really can't be tested or really can't be answered. So that makes them not testable. So these are religious questions or questions that are based on faith. Questions that surround supernatural events. Sometimes this could be considered religious, sometimes not. There are some things that you just can't answer because you can't test it. Another really important thing about a hypothesis is that you never prove them. You support a hypothesis. So especially in our early labs, one of the biggest point losses that I take off is that students say, I proved it. I proved my hypothesis correct. You did not prove it. You supported it. Your data suggests that your hypothesis is a plausible explanation. The reason that we say that it's supported and not proven is that you cannot test your hypothesis in every single situation. You might be able to prove it if you could test it in every situation that this would come up, but you can't. And so we say that we have supported our hypothesis, or on the opposite end, that we did not support our hypothesis. Our data did not support our plausible explanation. Once we form a hypothesis, we can then go about making our experiment and designing a way to test our experiment. So we formulate our hypothesis, we set up our experiment using our control group, our experimental group, our dependent and independent variables. And once we have an idea of how we're going to test our hypothesis, we can then make a prediction. So a prediction is just a statement of the data that you think you need to support your hypothesis. Just like a hypothesis, your prediction might not be correct, but you're basing it on the information you already know. So a hypothesis is a general statement trying to explain or answer your question or your observation. Your prediction is the actual data. So your prediction usually has to include some sort of numbers or values that you are going to collect. You write this in the form of an if-then statement. And I'm not going to do an example right now because we're going to do an example in our lab, but I want you to kind of think about how this might look. So you're going to say, if your proposed explanation then numbers or values or data. So your hypothesis should not contain numbers, value, or data. It's a statement that's providing an explanation. Your prediction is associated with the experiment and the data that you should expect. Now, these take time to learn how to write correctly, and often students will just put an if and a then into their hypothesis and call it good. But when we get to this lab, and we'll do this in many of our labs, make sure that you're not including data in your hypothesis, but you're including it in your prediction, where you try to guess what's going to happen in your experiment. So we've asked a question, we've designed an experiment, we've made a hypothesis, we've predicted what data we expect to get, and we're going to actually do the experiment. Now, some experiments are really simple, some experiments are really complicated, and that's okay. It just depends on what you're actually testing. But at some point you have to gather data, and it might be a little data, it might be gobs and gobs of data. But the data that you record are what's going to help you decide if you have supported or not supported your hypothesis. So data is simply a set of recorded observations. And these come in two types. Qualitative data are descriptions. So this might be something like bigger, darker, less. Whereas quantitative are actual values, data that comes as a value. So these are going to be usually numbers, a measurement of some sort, and either one of these are equally valid forms of data. Usually you want to shoot for quantitative, and the reason for that is that it's a little bit easier to make distinctions. If you want to say something's bigger, well, what do you mean by bigger? That's completely subjective. So sometimes qualitative, while not useless, are not the best way to collect our data. But if we have quantitative values, if we have a specific measurement, we can set, what do we mean by bigger? Well, we mean twice as big, or we mean 5 inches longer, or we mean a few centimeters bigger. Whatever your data might be, it's easier to analyze it when you use quantitative values. And one of the ways that we can analyze those quantitative values is by using statistics. So statistics are going to tell us how likely our outcome is. Now, we won't do a lot of statistics in this class. We will do the chi-squared analysis, and we'll do that in our genetics unit. And that's just a way for us to determine, like, at what point do we say, yes, this data is different. Our experimental value is different than our control value. Once we have our values, there's a lot of different ways that we can represent it. We're going to make some different types of graphs in this class. You can represent your data using tables, graphs, charts, whatever is appropriate. And we'll do some of those things as we go through our labs. So the last step in the scientific method is basically drawing conclusions about your data. You collected your data, you analyzed it, you maybe did some statistics on it, and the last thing you need to do is evaluate whether or not you have supported your hypothesis. Remember, we don't prove a hypothesis, but we can support it or we cannot support it. And both supporting and not supporting our hypothesis is equally valid. You're not wrong if you have not supported your hypothesis. It's okay to not support a hypothesis. But whether you support or do not support your hypothesis, you want to think about what you learned in that experiment. Did you support it? What does that tell you about the system? What can you learn from this new understanding, this new causal relationship between an experimental variable and what actually happens? You also need to think about further studies. What could you actually do now? What other explanations might you need to look for? If your hypothesis was not supported, why do you think it wasn't supported? Could you do a different test? Could you do a little more research and see if a different hypothesis might be more likely? And you also need to think about what were your experimental problems and your limitations. So sometimes we have uncontrolled variables. Now, uncontrolled variables are things that you as the researcher have no control over. Now, what these are depend greatly on the experiment that you're designing. And sometimes you might just have to say, you know what, I can't control that variable. I know that it might affect my experiment, so I just have to keep that in mind. Other times, you can actually set up the experiment to limit those uncontrolled variables or those outside factors. And you do that by keeping trials consistent between your control and your experimental. You want as many things to be the same as you can. Uncontrolled variables are not necessarily bad, but you just have to evaluate how much you think they're affecting the experiment, because sometimes uncontrolled variables can have a great effect on your experiment. So we really want to try to limit the uncontrolled variables that we have in our experiments. It's also really useful to identify the limitations. What couldn't you test? Why couldn't you test it? And often in our labs, our lab manual is going to ask, did you support your hypothesis? Why or why not? And did you have any problems? And what we're looking for in this situation is experimental design problems. So we are looking at the limitations of the experiment. Experimental design. Now, a lot of times students like to say, well, I didn't do it right. And that is not a valid response here. When you're going through and doing our experiments, you should understand what you're supposed to be doing so that you don't mess it up. Sometimes things happen, it's okay. But what we don't want to include here is, well, I didn't read the instructions, I didn't understand it, I did it wrong. You need to do the experiment correctly. You need to understand the process of the experiment so that you know that the experiment is done correctly and that any limitations are due to the way that you designed the experiment. Maybe you didn't quite have the right equipment. Maybe the equipment you had wasn't sensitive enough. Things like that. And then sometimes you just have crazy data that just doesn't really make sense, and just do your best to figure out, what can I learn from this? Were there problems with the experiment? What were the limitations there? So the last thing for this lecture is discussing the difference between a theory and a hypothesis. So we just talked about a hypothesis. It is a plausible explanation to a question. A theory, in contrast, is an explanation for a wide range of events. In other words, it's a very, very broad explanation that covers a whole bunch of different situations in which little hypotheses, little experiments supported those little hypotheses. And all of those hypotheses together, the same sort of hypothesis is supported over and over and over again to provide a large body of evidence to say, hey, it seems like, in air quotes, every situation, this happens. Now, it's not happening in every situation necessarily, but when you have a theory, it's supported by a very large body of evidence. So some examples of this are Darwin's theory of evolution. Darwin didn't go out and look at one little organism and say, oh, look, it changed over time. He took a whole bunch of evidence that has since been corroborated and repeated, and new evidence found that still fits and supports his theory of evolution. There are lots of other theories in biology and other sciences that are supported by a wide body of evidence. Sometimes in our sort of everyday language, you use theory as more of a hypothesis. I have a theory about that. And what you're really talking about is a little explanation to one situation. So theories are ideas that we shouldn't really take lightly because they have been supported by a wide body of evidence. Now, a theory, just like a hypothesis, can be falsified. So you may not support a theory in every situation. And a theory might need to be revised if we see that continuously, that theory is not supported in new situations. So just sort of as a reminder or in contrast, a hypothesis is much smaller than a theory. It explains a very specific set of parameters, a very specific experiment. And that hypothesis might apply to other situations, but your hypothesis is very narrow, whereas a theory is very broad. Keep in mind that we are always looking to support the hypothesis and support the theory. We're not proving them. And likewise, both of them can be rejected, or we could not support it. And then we need to evaluate, why are we not supporting it? Are we not supporting because it's actually not supportable in a wide range of situations, or are we just not supporting it? Is there a different explanation in this situation? So in the textbook, section 1.3, it goes through the scientific method. And as a really cool example, they talk about mouse color and the environment on sandy versus light colored sand versus dark colored sand. And it was this really cool experiment that was attempting to provide a hypothesis for the explanation for two different coat colors in these little mice that live on the sand dunes. So I would strongly suggest that you read through that section. It's not very long. Take some notes, and maybe along with your notes, use this experiment to fill in some examples of what is the hypothesis in the experiment? What might the prediction be? What data did they gather? How did they support their hypothesis?